Hello everyone, I hope you are doing well. In this session I'm going to talk about excitable skeletal muscle cells. As you see, this picture shows the neuromuscular synapse, also known as the end plate, is the junction where a motor neuron communicates with the skeletal muscle fiber. This site is crucial for transmitting signals from the neuron system to initiate muscle contraction. A neuron acts as the presynaptic cells that send signals to the muscle. When an action potential travels down the motor neuron, it triggers the release of neurotransmitters, primarily acetylcholine into the synaptic cleft. In the neuromuscular synapse, I told you we have neurons and the skeletal muscles. The post-synaptic cell is a skeletal muscles cell that receive the signal. Acetylcholine binds to the receptors on the muscle cell membrane, leading to depolarization and the initiate of an action potential in the muscle fiber. We have an important process that's named the excitation contraction coupling. This process links the electrical signals from the neuron to the mechanical contraction of the muscles. The action potential travels along the muscle fibers membrane and into the T-tubules and triggering the release of calcium ions from the sarcoplasmic reticulum. Calcium binds to troponin and causing a shift in tropomyosin and exposing bonding site on actin for myosin heads, leading to muscle contraction through the sliding filament mechanism. Okay, as a summary, the interaction between neurons and skeletal muscle fiber at the neuromuscular junction is essential for voluntary muscle movement. The precise coupling of electrical stimulation and mechanical contraction allows for coordinated muscle action, critical for activities ranging from simple movement to complex motor tasks. Okay, here is an overview of excitatory postsynaptic potentials, EPSP, as a local potential, and their relationship to action potentials, including the concepts of spatial summation and temporal summation. The excitatory postsynaptic potential, EPSPs, are small depolarizations of the postsynaptic membrane that occur when excitatory neurotransmitters bind to receptors on the postsynaptic neurons. A single EPSP, typically around 0.5 to 1 millivolt, is insufficient to trigger an action potential on its own due to its small magnitude. Okay, an action potential is I told you in a before session, is a rapid, large change in membrane potential that propagates along the neurons, allowing for the transmission of signals. An action potential is generated when the membrane potential reaches a certain threshold, okay, typically around minus 40 to minus 55 millivolts. There are key phenomena leading to action potential: spatial summation and temporal summation. Spatial summation occurs when multiple presynaptic terminals simultaneously release neurotransmitters, leading to several EPSPs that can add together. If the combined effect of these EPSPs is sufficient to reach the threshold, an action potential will be triggered. But the temporal summation occurs when a single presynaptic terminal fires multiple times in rapid succession, causing successive EPSPs. If the timing and frequency of these discharges are appropriate, the cumulative effect can lead to a depolarization that reaches the threshold for an action potential. Okay, both spatial and temporal summation are critical for the generation of action potentials in neurons. They allow for the integration of multiple signals, ensuring that only significant stimuli result in the firing of an action potential, thus facilitating effective communication within the nervous system. Okay, as you see, this picture shows the excitatory postsynaptic potential, the blue and the green curve, and the action potential show in red curve. Okay, this curve, the x-axis is time and y-axis is voltage, and show when the for example the few synapses 4 or 8 synapses fire will not cause sufficient summated potential to elicit an action potential, but the simultaneous firing of many synapses, 16 synapses, will raise the summated potential to threshold for excitation and cause a superimposed action potential. Okay, here is an overview of the release of acetylcholine from the presynaptic neuron terminal and its interaction with receptors in the postsynaptic muscle cell. When an action potential reaches the presynaptic terminal of a motor neuron, it triggers the opening of voltage-gated calcium channels. Calcium ions enter the neuron, leading to fusion presynaptic vesicles containing acetylcholine with the presynaptic membrane. And the acetylcholine is released into the synaptic cleft through exocytosis. Acetylcholine diffuses across the synaptic cleft towards the postsynaptic muscle cells. You must know the primary receptors on the postsynaptic muscle cells are nicotinic acetylcholine receptors, because we have two type of the receptors for acetylcholine, muscarinic and nicotinic. In this area, in the muscle cells, we have a nicotinic acetylcholine receptors. Acetylcholine bind to these receptors, causing a conformational change, and the binding of acetylcholine, these receptors opens ion channels in the receptors, allowing the sodium ions to flow into the muscle cells. These influx of sodium ions lead to depolarization of muscle cell membrane and generating an excitatory postsynaptic potential, or EPSP. If this depolarization is sufficient to reach the threshold, an action potential is generated in the muscle cells and leading to muscle contraction. As a brief, the release of acetylcholine from the presynaptic neurons terminal and its bonding to receptors on the postsynaptic muscle cells is a critical step in neuromuscular transmission. This process facilitates the conversion of a neurosignal into a mechanical response, enabling muscle contraction and movement. And you must know the botulinum toxin reduce the release of acetylcholine from the neural terminal. As I said, that botulinum toxin is a potent neurotoxin produced by the bacterium Clostridium botulinum. It is known for its ability to inhibit the release of acetylcholine at the neuromuscular junction. Botulinum toxin enters the presynaptic neuron and cleaves proteins essential for the fusion of synaptic vesicles containing acetylcholine with the presynaptic membrane. This prevents the exocytosis of acetylcholine in the synaptic cleft, leading to reduced communication between the neuron and the muscle. The reduction in acetylcholine release results in decreased muscle contraction, which can lead to temporary paralysis of the affected muscles. This property is exploited in medical treatment, such as for muscle spasticity, chronic migraines, and cosmetic procedure. It is very important because especially for women, because this action reducing wrinkles. Botulinum toxin effectively reduces the release of acetylcholine from presynaptic neurons, resulting in decreased neuromuscular transmission and muscle paralysis. This mechanism underlies both its therapeutic applications and its potential dangers. The action potential is skeletal muscle cells, including key details such as resting potential, threshold, and the phase of action potential. Similar to the another action potential. So in the resting potential in the skeletal muscle cells is approximately minus 80 millivolts, and similar the resting membrane potential in another excitable cells. The resting membrane potential is the electrical charge across the muscle cell membrane when the cell is not actively sending a signal. This is maintained by the distribution of ions, mainly sodium and potassium, across the membrane. As a brief, the resting membrane potential is a voltage difference between inside and outside of the cell. And the threshold in the skeletal muscle is approximately minus 40 millivolts. I told you threshold is the critical level of depolarization that must be reached for an action potential to occur. If the membrane potential reaches this threshold, voltage-gated sodium channels open, leading to rapid depolarization, and the similar to the another action potential in the muscle cells have three phases. The depolarization, upon reaching the threshold, the sodium channels open and sodium ions rush into cell. This influx of positive ion causes the membrane potential to become less negative and eventually positive. Sometimes we have one phase that's named overshoot. The potential exceeds zero, okay? After the stimulation, when the potential exceeds zero, often reaching around plus 30 millivolt, this phase named the overshoot. And this phase represents the peak of the action potential, where the interior of the cell is positively charged relative to the outside, more than to the zero, okay? When the voltage of these cells after the stimulus exceed from zero and reach to the positive amount, this part of action potential called overshoot. After that, sodium channels closed and potassium channels full opened, allowing potassium ions to exit the cell. This outflow of positive ions causes the membrane potential to return toward the resting potential. This phase is repolarization, okay? So, sometimes the membrane potential temporarily becomes more negative than the resting membrane potential, often dropping to about minus 90 millivolt or minus 100 millivolt, and this occurs due to the continued efflux of potassium ions before the potassium channels close, because I told you the kinetic of the potassium channels is slowly. So making the cell less likely to fire another action potential immediately. So as a whole, the action potential in skeletal muscle cells is a rapid and coordinated change in membrane potential that allows for muscle contraction. In this picture, again, explain the coupling of stimulation with contraction in a skeletal muscle. As you see, the action potential generated in the muscle cell membrane travels along the sarcolemma, muscle cell membrane, and into the T-tubule, which are invagination of the membrane, These electrical signals triggers the subsequent steps leading to muscle contraction. This action potential stimulates the sarcoplasmic reticulum, an organelle that stored calcium ions. These depolarization caused by the action potential results in the opening of calcium channels in the sarcoplasmic reticulum, leading to a rapid increase in intracellular calcium concentration. The increase in calcium concentration allows calcium to bind to troponin, a regulatory protein on the actin filament. This bonding causes a conformational change in tropomyosin, another protein that blocks the bonding site on actin, and with the bonding sites exposed, myosin head can attach to actin and forming cross bridge. The myosin heads pivots, pulling the action filaments toward the center of the sarcomere, resulting in muscle contraction. As the actin and myosin filaments slide past each other, the length of the sarcomere decreases, leading to overall muscle shortening and contraction. The coupling of stimulation with contraction in a skeletal muscle involves the propagation of action potentials, a significant increase in intracellular calcium concentration from the sarcoplasmic reticulum, and the sliding of actin and myosin filaments. This coordinated process results in the shortening of the sarcomere and the contraction of the muscle. Okay, skeletal muscle is composed of specialized cells and structures that enable contraction and movement. The muscle fiber cells, also known as sarcolemma, these are the individual muscle cells that make up skeletal muscle. Each muscle fiber contains 100 to 1,000 of myofibrils, which are the contractile elements of the muscles. Myofibrils are made up of contractile proteins, primarily actin, thin filaments, and myosin thick filaments. In the characteristics of the myofibril, we have two bands, light bands or I bands and dark bands or A bands. The light bands contain only actin filaments. They appear lighter under a microscope, and the dark band or A band contain both myosin filaments and the overlapping ends of actin filaments. They appear darker due to the presence of both types of filaments. And we have a Z line in this structure. The Z line or Z disc are the boundaries of each sarcomere to which the ends of actin filaments are attached from both sides. Z-line help maintain the structure integrity of the myofibril. Another structure in the muscle is sarcomere. The sarcomere is the basic contractile unit of skeletal muscle, defined as the section of the muscle fiber located between two Z lines. Sarcomeres contract and shorten during muscle contraction, leading to overall muscle movement. The structure of the skeletal muscle is complex, consisting of muscle fiber cells, sarcolemma, myofibrils, and contractile filaments, actin and myosin. Okay, the arrangement of these components into light bands, dark bands, Z-line, and sarcomeres allows for efficient contraction and movement. This picture shows the organization of proteins in a sarcomere. Each titin molecule is extended from the Z line to M line. Part of the titin molecule is closely associated with the myosin-thick filament, whereas the rest of the molecule is springy and changes length as the sarcomere contracts and relaxes. As a more explain, you must know the M line is a structural component found in the center of the sarcomere, which is the basic unit of muscle contraction. It serves as an anchoring point for thick filaments, myosin, and helps maintain the alignments of the sarcomere during muscle contraction. But the titin is a large protein that spans half of the sarcomere, connecting the Z line to the M line. The titin provides elasticity and stability to the muscle, allowing it to stretch and recoil, and it helps maintain the structure of the sarcomere and plays a role in muscle contraction and relaxation. As a whole, the M line and titin are crucial for the structural integrity and the functionality of muscle fibers, ensuring efficient contraction and relaxation during muscle activity. In this slide, I am going to talk about the actin filament skeleton and associated proteins. As you know, the actin filament skeleton is a sequence of actin molecules, G-actin, that appears as a double helical structure that's called F-actin. And the tropomyosin is a protein that's wraps around the actin filaments, covering the active sites on actin. This prevents myosin from bonding to actin in a relaxed muscle state, regulating muscle contraction. We have three types of the troponin. Troponin 1, troponin T, and troponin C. The troponin 1 binds to actin, anchoring the troponin complex to the actin filament, and troponin T binds to tropomyosin, helping to position the troponin complex on the actin filament. And the troponin C binds to calcium ions. When calcium binds to troponin C, it causes a conformational change that moves tropomyosin away from the active sites on actin, allowing muscle contraction to occur. As a whole, the actin filament skeleton, along with tropomyosin and troponin complex, play a vital role in a muscle contraction by regulating the interaction between actin and myosin. As you see in this picture, show the actin filament composed of two helical standards of F-actin molecule and two standards of tropomyosin molecule that fit in the grooves between the actin strands attached to one end of each tropomyosin molecule in a troponin complex that initiates contraction. Okay, myosin has two heavy changes that spiral around each other, forming a coiled structure. These changes contribute to the formation of the myosin body, providing structure, integrity, and strength. The tail of the myosin is crucial for forming the myosin body, allowing multiple myosin molecules to aggregate and form thick filaments. And the arm extended from the tail and end in protruding heads, which are essential for forming cross bridges during muscle contraction. The movement of these heads is often referred to as gear movement, allowing the myosin to interact with actin filaments. The light changes of myosin are associated with the myosin head. These light changes play a role in stabilizing the head region and are involved in the regulation of the myosin activity. As a whole, the structure of the myosin, including its heavy changes, tail, arms, protruding heads, and light changes, is essential for muscle contraction. Okay, in this picture show the sarcomere contraction. The shortening of the sarcomere length is synonymous with muscle contraction. During contraction, the actin filament slides over the myosin filaments, leading to a reduction in the overall length of the sarcomere. Essential components for contraction is calcium. Calcium binds to troponin C, causing a conformational change that movement tropomyosin away from the active sites on actin. This exposure allows myosin heads to attach to actin, initiating the cross-bridge cycle. Another component that's essential for contraction is adenosine triphosphate, or ATP. ATP is required for several steps in the contraction process. It provides the energy needed for the myosin heads to detach from actin after a power stroke, and it is also necessary for the recocking of the myosin heads, preparing them for the next cycle of contraction. Without ATP, myosin head cannot release from actin, leading to a state of rigor or rigor mortis. As a whole, the contraction of sarcomere resulting in muscle shortening is dependent on the presence of calcium ions and ATP, which facilitate the interaction between actin and myosin filaments. In this picture show the effect of sarcomere length on contractile strength. Length tension relationship between the length of a sarcomere and its ability to generate force is known as the length tension relationship. In the optimal sarcomere length, typically around 2 to 2.2 micrometers in human muscle, where the overlap of actin and myosin filaments is ideal. At this length, the maximum number of cross bridges can form, resulting in the greatest contractile strength. But in the shortened sarcomere length, when the sarcomere is too short, less than the optimal length, overlapping the actin filament can interfere with myosin heads' ability to bind, leading to a decrease in force production. These filaments may become crowded, limiting effective cross-bridge formation. When the sarcomere is too long, in the elongated sarcomere length greater than the optimal length, there is insufficient overlap between actin and myosin, resulting in fewer cross bridges and a decrease in contractile strength. Myosin heads cannot effectively engage with actin, and diminishing force generation. As a whole, the contractile strength of a sarcomere is highly dependent on its or sarcomere length. Optimal sarcomere length allows for maximum cross-bridge formation, while deviation from this length, either too short or too long, result is decreased contractile strength. In this slide, define the motor unit. Motor unit consists of a single motor neuron and all the muscle fibers it innervates. This unit is the fundamental component of muscle contraction. We have two type of the motor units according to the size: a small motor unit and large motor unit. The small motor units found in the muscle responsible for precise movements. Laryngeal muscles, eye muscles, these units consist of fewer muscle fiber, allowing for fine control. But the large motor unit found in muscles that generate greater force, quadriceps muscles. These units consist of many muscle fibers, enabling powerful contractions but less precision. Muscle fibers can be classified into several types based on their characteristics, functions, and metabolic properties. The main types include type 1 fiber or slow twitch fibers. These fibers are high endurance and fatigue resistance, rich in mitochondria, myoglobin, and capillaries, giving them a red appearance and a slow contraction speed. Primarily used for aerobic activities and endurance exercise, e.g. marathon running, long distance cycling. But type 2 fibers or fast twitch fibers are further subdivided into two main categories. Type 2A fiber or fast oxidative fiber. This fiber intermediate endurance and fatigue resistance, contain a moderate amount of mitochondria and myoglobin, and faster contraction speed than type 1 fibers. This fiber is suitable for activities that require both strength and endurance, e.g. middle distance running and swimming. Type 2B fibers, fast glycolytic fibers, is low endurance and high fatigue rate, fewer mitochondria and myoglobin, giving them a white appearance, and it is a very fast contraction speed, and this fiber designed for short bursts of power and strength, e.g. weightlifting. As a whole, the muscle fibers are classified in two types: one, slow twitch, and type 2, fast twitch fiber, with type 2 further divided into type 2A and 2B. Each type has distinct characteristics and functions, catering to different physical activities and performance needs. We have two types of muscle contraction based on how the muscle fibers generate force and change length: isometric contraction and isotonic contraction. Isometric contraction, in this contraction, the muscle generates force without changing length. Tension increases, but there is no movement of the joint. Commonly occurs during static exercise, e.g. holding a weight in place. For example, when you pushing against a wall. In the isotonic contraction, the muscle change length while the tension remain constant. We have three concepts about the muscle size or growth: atrophy, hypertrophy, and hyperplasia. In the atrophy refers to the decrease in muscle size and strength due to a reduction in muscle fiber size or number. The cause of the atrophy can be lack of physical activity or immobilization, for example, after injury, or inadequate nutrients intake, and finally conditions such as muscular dystrophy or aging. But hypertrophy is the increase in muscle size and strength due to an increase in the size of muscle fiber. The hypertrophy can be happen when the regular weight lifting or resistant exercise stimulate muscle growth, or increased level of anabolic hormones such as testosterone, growth hormone promotes hypertrophy. The other the concept is hyperplasia. Refers to an increase in the number of muscle fiber. This phenomenon is less common in skeletal muscles but can occur in a specific condition. For example, in response to iodine deficiency, the thyroid gland can undergo hyperplasia as it attempts to produce more thyroid hormone. Thank you for your attention. I hope you have studied the material up to this point from the book.